Hello children, in today's video number 4, I am going to deal with the topic immune disorders. The first immune disorder we are going to study is allergy. So let's define allergy. Allergy is the exaggerated response of the immune system to certain antigens present in the environment. So once again, what is allergy? It is defined as the exaggerated response of the immune system to certain antigens present in the environment. These antigens that are present in the environment are called allergens, and the allergens include dust, pollen grains, certain foods, animal dander. Animal dander means the fur of animals. And during allergic reactions, what happens is our immune system responds by releasing the antibody IgE. So why it is coming under immune disorder? Because the immune system is recognizing and getting activated by releasing the antibody IgE. And the allergic reactions include sneezing, eye watering, rash, bruises, running nose, etc. Now the cause of these allergic reactions are due to certain chemicals like histamine and serotonin released from the mast cells of the areolar tissue. So these allergic reactions, whatever may be the allergic reactions, these reactions are caused due to the chemicals released by the mast cells. The chemicals are histamine and serotonin. Detection of allergies. How do we detect that we are sensitive to a particular allergen in the environment? There is a test called the skin prick test or the puncture test, or it is also called the patch test. In this test, the forearm, the upper part of your forearm is cleaned with alcohol or a sanitizer, and on the skin with the help of a marker it is coded with numericals 1, 2, 3, 4, 5. Nearly 50 allergens are tested at a stretch. So your forearm is coded with a marker, skin marker, and each code stands for an allergen. Next, what do is with the help of a sterilized filler, they put a drop of the different allergen besides each code. If 1 is indicating pollen, 2 is indicating pet dander, 3 is indicating dust, 4 is indicating pollen, chalk powder. Against each numerical, they add a drop of the allergen. Next, what do is, through the drop with the help of a sterilized lancet needle, they prick such that the allergen penetrates down into the dermis. Once it penetrates down to the dermis, whichever the person is sensitive to, it produces an allergic reaction. How we will understand this allergic reaction? The patient should wait for 30 minutes. After 30 minutes, whichever allergen he is suspected to be sensitive shows a red, raised, itchy bump with a red ring. That is called wheal, W-H-E-A-L. And then the skin specialist identifies which code he is allergic to. It's number 4. So he is allergic to, as I told you in the beginning, number 4 corresponds to chalk powder. Or if it's number 1, he is allergic to dust. Or number 2, he is allergic to animal dander. Against whichever code he checks with the allergen, and the sensitivity to that particular allergen is determined. So this is the way allergies are being determined. The patient is exposed to small amounts of the allergens by the skin prick test. I have taken a picture of the skin prick test. See, you can see the forearm where drops of allergens are placed and pricks are made, and finally after 30 minutes this wheal appears, WHEAL, against the substance he is allergic to. So, this is the way how skin allergies allergies are detected. Now, the treatment of allergies, the better way to get rid of allergies to avoid that particular substance. Once you identify that you are allergic to a particular foodstuff or allergic to pollen grains or allergic to dust, avoid. If at all you cannot avoid such scenarios, the only measure of treatment is by taking certain drugs which are containing antihistamines, adrenalins and steroids. These work by reducing the effect of the allergic reaction. You will not be completely free of the allergic reactions. The allergic reactions will be highly reduced or significantly reduced. So, once again, what are allergies? Allergy is the exaggerated response of the immune system to certain substances or antigens present in the environment. These substances or antigens present in the environment are called allergens, and allergens includes pollen grains, dust, pet danders, chalk powder, etc. And the allergic reactions include sneezing, running nose, rash, etc., etc. And how the immune system respond? They release the antibody IgE. We know there are 5 antibodies. The antibodies that is released during the time of allergy is IgE. Then the cause of this allergic reaction is due to the chemicals released by the mast cells, namely histamine and serotonin. Detection of allergy is by the skin prick test or the patch test or the puncture test, where the patient is exposed to small amounts of different allergens. And I gave you a brief procedure of the skin prick test. It is not there for your NCERT, I just gave you as an extra information. Anyway, it is by exposing the person to small amounts of allergens that the doctor identifies to which allergen the person is sensitive to. And the measure is to avoid that particular substance. But if we cannot avoid, then in that case we can go for treatment, and the treatment involves taking in antihistamines, adrenalins or steroids. So, that is all related to allergies. Next, immune disorder. Autoimmune disorders. We know the role of the immune system. The immune system, the role is to defend the body against diseases. But sometimes the immune system goes off the track, and instead of defending us from diseases, it attacks the self cells. Instead of identifying self cells from non-self cells and protecting the self cells from antigens, what happens is the immune system does not do its role. It goes off the track and attacks the self cells. In that case, it results in autoimmune disorders. So, the definition is like this: the condition in which the body attacks self cells, resulting in the damage of the body. So, the immune system, instead of protecting the body, it attacks the body. The reason may be, usually it is genetic or sometimes due to some unknown reasons. It may not be genetically inherited. The best example is rheumatoid arthritis. I have given the picture of a rheumatoid arthritis, the hand of a person having rheumatoid arthritis. The characteristics of this particular disease is that the joints become very stiff and very painful and also very disfigured. Look, a disfigured hand. The reason is that the immune system mistakenly sends antibodies to the lining of the joints. We know the articulating bones. A joint is a junction between 2 or 3 bones, and the 2 bones are surrounded by a layer called synovial tissue, and there is a fluid here. Okay. Now, what happens is that the immune system sends antibodies, and these antibodies attack the synovium. What is synovium? The membrane surrounding the articulating surface of the bones. And what happens is that they become this synovium covering the joint or covering the articulating bones becomes sore, inflamed, and release certain chemicals that cause damage to the nearby bones. So, once again, rheumatoid arthritis, in this condition what happens, the immune system mistakenly send antibodies where? To the joints. And what happens? These antibodies attack the synovium. What is synovium? The layer of tissues surrounding the articulating bones of the joint. And what happens to these this layer or synovium? This covering sews up, inflames and release chemicals, and these chemicals attack the adjacent bones, and therefore the stiffness, pain, inflammation, and finally disfigured joints. This is rheumatoid arthritis, one autoimmune disorder. There are a number of autoimmune disorders, but the one which is mentioned in the NCERT is the rheumatoid arthritis. So, once again a recap of autoimmune disorders. The immune system, the role of the immune system is to defend our body against diseases. But sometimes the immune system goes off the track and attacks the body cells. Such a situation is known as autoimmune disorders. It may be genetical or may be due to some unknown reasons. Example is the rheumatoid arthritis, disfigured, painful, inflamed, stiff joints. It's not due to aging. We know about arthritis caused at the time of aging after osteoporosis, that is after menopause. Usually many females develop arthritis. Not that arthritis, this can attack even youngsters because it's an autoimmune disorder. The immune system mistakenly sends antibodies that attack the synovium. Synovium is that layer protecting the articulating surface of the bones at the joints. And what happens to the synovium? It sews, inflames and releases chemicals that attack the bones. I hope autoimmune disorders are clear. Next we have the popular immune disorder, AIDS. It's acquired immunodeficiency syndrome. And what is this condition? It's a condition in which the person becomes immunodeficient in such a way that the body is unable to defend itself against disease. So, AIDS is a condition in which the person becomes immunodeficient, that the body is unable to protect against infection. Body means the immune system is unable to protect the body against any infections. Even the slightest cold he is susceptible to fall into. Now, it is caused due to decrease in the number of helper T cells, significant decline in the number of helper T cells. When we learn the replication of the retrovirus, we can see how the number of helper T cells gets declined. Now, mode of transmission, we have learnt in chapter 4, there are 4 different modes of transmission of AIDS. One is from the infected mother to the fetus. 2, from unsterilized syringes. Or medical equipments. 3, from blood transfusion. And 4, sexual contact. So, these are the 4 methods or modes of transmission of the virus that cause AIDS, that is from the infected mother to the fetus due to the use of unsterilized syringes or medical gadgets, then blood transfusion, and finally due to sexual contact. The pathogen is HIV, it stands for human immunodeficiency virus. The detection test is called the ELISA test. E stands for enzyme, L stands for linked, I stands for immuno, S stands for sorbent, A stands for assay. Enzyme, linked, immunosorbent, assay. Then the treatment. Actually, there is no treatment for AIDS, but we can administer antiretroviral drugs. It can just only prolong the life of the person, but death is inevitable. He cannot avoid death. Just for some time we can prolong the patient's life by taking antiretroviral drugs. Anyway, there's no complete cure. There's no cure to AIDS as of now. So the only measure is prevention. And I told you the famous ABCD in chapter 4. A stands for avoid sex with multiple partners or unknown partners. A, avoid sex with multiple partners or unknown partners. B, the best measure. Be faithful, so you will not fall into this. C, use condoms so that you will not contract this disease or any other STDs. And D, if at all you are having a doubt that you are having AIDS, go to the doctor and get yourself detected. So here it incorporates 3 Ds. If you are doubtful, go to the doctor and get yourself detected. And in this context, to bring down the spread of AIDS, the WHO, the NACO and a number of NGOs are taking a lot of measures to bring down the spread of AIDS. There are lots of awareness programs, educating the masses, then giving condoms free of cost, then checkups, periodic checks to those population that are susceptible to HIVs, all free of cost, and also awareness programs related to advocating safe sex, etc. So all these organizations have been working towards bringing down the spread of AIDS, and that has to some extent been successful. So once again, a recap. What is AIDS? AIDS is a condition in which the person becomes immunodeficient, that the body is unable to protect against infections. It is caused due to the decrease in the number of helper T lymphocytes. The mode of transmission are 4. We have learnt in the 4th chapter. 1 is from the infected mother to the fetus, 2 from unsterilized syringes, 3 from blood transfusion, and 4 from sexual contact. The pathogen is HIV, human immunodeficiency virus. The detection test is called ELISA test, enzyme linked immunosorbent assay, and there's no cure. Remember, there's no cure for AIDS, but if the person is having AIDS, there's treatment just to prolong his life. Death is inevitable and he suffers of multiple infections because the immune system is very, very low or zero, that he gets prone to any disease which otherwise the person can overcome. Even the disease by the simplest bacterium, mycoplasma, or by a protozoan like toxoplasma. Even simple infection, simple flu, he is susceptible to get the attack. But the treatment is by administering antiretroviral drugs. And lastly, prevention, and here I wish to emphasize the adage. Prevention is better than cure, so follow the ABCDs. A, avoid sex with multiple partners or unknown partners. B, be faithful. C, use condoms. And D, if you are doubtful, go to the doctor and get yourself detected. Now let's go into the life cycle or the replication of HIV. How do the HIV multiply once they get into the human body? Once the retrovirus gets into the human body, what happens? First of all, you must know the basic structure of a virus. It's very simple. It just has 2 parts, a protein coat surrounding it as well as a genetic material. Genetic material can be RNA or a DNA. If RNA is a genetic material, it's called retrovirus. And if DNA is the genetic material, they are the other type of viruses. So we have 2 types of viruses. The difference between the viruses, either they have RNA or DNA as a genetic material. But all viruses are surrounded by a protein coat. That is common to all viruses. So the structure is very simple. So we have the retrovirus, it invades the host. Once it gets into our body through any of the modes of transmission, it attacks the cell. Which cell? The macrophage. So remember, and you have to reword or write in your text against this box which is indicated in the box, that cell is the macrophage. It invades the macrophage. Once it invades the macrophage, see it's entering. What enters is the RNA, viral RNA enters, leaving behind the protein coat. So what enters the macrophage is the viral RNA. The protein coat is left behind. Now this viral RNA is inside the macrophage. Now before going further, I want to bring a small topic of genetics here. The central dogma of molecular biology. It is like this. From DNA we get RNA, and from RNA we get proteins. This is called the central dogma in microbiology. So the process by which RNA is derived from DNA is called transcription. And the process by which from the RNA we get the protein is called translation. Now we are only bothered of this portion. Transcription. So what is transcription? The process by which an RNA is formed from the DNA is called transcription. From DNA to RNA. Remember that. Now here we have the viral RNA. So I am incorporating this information here. Now the viral RNA has entered the host cell, that is the macrophage. Now what happens is, this is viral RNA. This viral RNA becomes viral DNA. So this is RNA, viral RNA becomes viral DNA. So which is the process? From DNA to RNA it's called transcription. So from RNA to DNA it's the reverse. So what will be the process called? Reverse transcription. And this is helped by an enzyme called reverse transcriptase. So once again the viral RNA that has invaded the macrophage becomes the viral DNA with the help of the enzyme reverse transcriptase. So we got the viral DNA from the viral RNA, and we know the difference between RNA and DNA. RNA is single stranded, DNA is double stranded. RNA has what and all bases? AUGC. Here it is ATGC. So there are lots of differences between RNA and DNA. But here remember, it is not DNA to RNA, it's RNA to DNA. So definitely it's reverse transcription brought about by the enzyme reverse transcriptase. So this is the viral DNA. Now this viral DNA, see where's the arrow going? It's getting into the nucleus of the host. And we know the property of the nuclear membrane, it has pores. So through the pores it enters the nucleus. So this viral DNA gets into the nucleus. How? Through the nuclear pores. And it gets incorporated. Once it enters the nucleus, it gets incorporated into the host genome. The host is having DNA. So now this viral DNA gets incorporated into the host DNA. So we get host DNA plus the viral DNA. Okay, got incorporated. And then what happens is this process. From DNA RNA synthesis, transcription takes place. The host will undertake transcription. When transcription takes place, the DNA is converted to RNA. So here we can see the viral DNA gets transcribed into viral RNAs. So here normal transcription is taking place, and this viral RNA comes out through the pores out into the cytoplasm. So I got back the viral RNA. Like that way transcription is occurring. A lot of this transcription is taking place. So number of viral RNAs are synthesized and coming out through the nuclear pores. Okay, the role of the DNA inside is for transcription, and transcription is going on. When transcription is going on, what is happening? Viral DNAs are getting transcribed to viral RNAs, and these viral RNAs come out through the nuclear pores out into the cytoplasm. So you see, I have how many viral RNAs? 1, 2, 3, 4. And then what happens? There are the raw materials for the protein coat. They get surrounded. These viral RNAs get surrounded by the protein coats. So what we get back? We get the virus back. So in effect, the macrophage acts as a factory, HIV factory. See, one viral RNA got in, and how many viruses are being produced? Many. So it acts like a factory for HIV. So the macrophage serves as a factory. Now this macrophage multiplies. Simultaneously, the HIV has entered the T lymphocytes. There also the same process is occurring. There also the virus multiplies, and the number of viral progenies, and these all come out. See, they all come out of the cell. All these viral RNAs, they came out of the cell. All are coming out of the cell. And what do? Is they attack the helper T lymphocytes. They attack the T lymphocytes which are there in the blood, and that's how there is a decline in the T lymphocytes. So once again, the life history or the life cycle or the replication of the HIV virus. So first you must know the structure of a virus, and that to a retrovirus, it has just a protein coat and RNA as a genetic material. It gets into the host cell. Here the host cell I mentioned is macrophage. How it gets? It does not get inside as a whole. The protein coat is left behind and only the RNA gets inside. The viral RNA gets inside. Immediately what happens is the viral RNA gets converted to viral DNA by the process of reverse transcription catalyzed by the enzyme reverse transcriptase. Next event, what happens? It gets into the nucleus through the nucleopores and gets incorporated with the host genome. See, host genome is black in color, viral genome I have indicated red in color. Now, it got incorporated into the host DNA. Now, the role of the DNA is, as I told you, DNA undergoes transcription. When it undergoes transcription, automatically this is also getting transcribed into a number of viral RNAs. So, it's getting transcribed into viral RNAs, and these viral RNAs are not inside the nucleus, they come out through the nucleopores into the cytoplasm. That way, one viral RNA came out, second, third, fourth, and a number of viral RNAs are coming out due to the transcription. And these viral RNAs are immediately surrounded by protein coats. See, I am surrounding each by protein coats. So, what do we get back? We got back the HIV virus. That way, we can see the macrophage serves as a factory of HIV, and these come out of the cell. And when they come out, they attack the helper T lymphocytes, and that's how there is a significant decrease in the helper T lymphocytes. As this happens, when the T lymphocytes progressively decreases, the patient, the person, he is not knowing he is having AIDS, he develops bouts of fever, diarrhea and weight loss. For no reason he gets fever on and off, then diarrhea and a weight loss. But still he is in the window stage, he is not aware that he is having AIDS. These are the initial symptoms, but still he is not realizing. He thinks it's a normal food poisoning or normal fever and takes medicines and gets rid of the particular symptoms. But after years and years, it results in chronic stage where his immune system becomes very weak or zero, that he is prone to any type of infections. He gets infections which otherwise normally he could have overcome. And finally, an AIDS patient is not dying of AIDS, actually he is dying of number of diseases where he cannot defend, his body cannot defend. He gets all the diseases and die a very painful death. Okay. So, what we should understand is to follow the ABCD. It's a very risky, fatal death, and to some extent there is a stigma attached to this AIDS, where the people do not accept us, though we innocently contract the disease or deliberately we get it due to our bad character, we are not easily acceptable in the society. So, better we take care and follow the ABCDs. Next, we are moving to the most dreaded disease. All are not been scared of AIDS, all are scared of cancer. It's just of course a dreaded disease, and many people have been dead due to this particular disease. But now with improvement in science and technology and medication and the early detection, now there are lots of cancer survivors, and now it has become more people have adjusted to this and they have taken up lightly and they have gone for early detection and treatment, and it has now that intensity of death due to cancer has declined, okay, because mainly due to the modern medicines and detection techniques. Anyway, what is cancer? So, let us make a comparison with the normal cells. So, this is the case of a normal cell. First property, normal cell growth. In normal cells, the cell growth and cell division is regulated, is controlled, well controlled or well regulated in normal cells, whereas when we make a comparison with cancer cells, this is uncontrolled. Cell growth and cell division is unregulated or uncontrolled in cancer cells. So, that marks the first difference between normal cells and cancer cells. And here I have a picture. See, normal cells, first division, 2 cells are formed. Second division, 2 cells are formed, and we know the division, 2 cells for each division means it's mitosis, and see, it stops here. So, it is controlled or regulated. But look here, divides into 2, then further divides, and see here uncontrolled growth, uncontrolled. A mass of cells are formed, okay, uncontrolled. So, the first property is uncontrolled growth of cells and division. Second difference, why this growth is controlled, cell growth and cell division is controlled? Because of a particular property called contact inhibition. See, now we saw the 4 cells that are formed after the second division are in contact with each other. See, they are in contact. By virtue of contact, they stop dividing. So, what is contact inhibition? The definition for contact inhibition is the cells by virtue of contact stops dividing just because they are in contact. Earlier they were not in contact. 2 cells that were formed are distantly located. Then these 2 cells divided, and now the daughter cells, see, they are just touching each other physically, just a touch, not overlap, just touch. So, that itself will switch off the mechanism. It regulates to stop the cell division, stop. So, this is called contact inhibition. So, what is contact inhibition? By virtue of contact of the cells, the cell division stops. Whereas, in cancerous cells, this contact inhibition is lost. The property of contact inhibition has been lost. See, though it is in contact, it continuously divides, redivides and overlaps and forms a mass of cells. And this mass of cells, see, continuously dividing, overlapping, contacting, but no contact inhibition, and it forms a mass of cell, and this mass of cell is known as tumor. I hope you have understood the 2 difference between normal cells and cancerous cells. So, once again, normal cells, cell growth and cell division is controlled or regulated, whereas in cancer cells, cell growth and cell division is unregulated or uncontrolled. Reason for this stands as a second point, because of contact inhibition. So, normal cell shows contact inhibition. What is contact inhibition? By virtue of contact, the cell stops dividing, whereas cancer cells has lost the property of contact inhibition such that they divide and redivide and form a mass of cells, and this mass of cell is called tumors. Now, here we are bifurcating. There are 2 types of tumors. So, the next slide will be the types of tumors. Tumors are divided into 2 types, benign tumors and malignant tumors. Now, let us analyze the difference between the benign tumors and malignant tumors. Benign tumors are confined to their original location. See, I have shown you a benign tumor in the thigh region. It is confined there, it does not spread. It is confined to that particular location, it does not spread. Whereas, malignant tumors are not confined, it spreads. Second point I already highlighted, benign tumors, they are localized and they do not spread, whereas malignant tumors spread. How? Via the blood, and that process by which the malignant tumors spread through the blood is known as metastasis. Clear? And the problem is that the cells, see, this is a malignant tumor, and the cell slaughter from this tumor is carried by the blood, and wherever they get deposited or wherever they get lodged, they form a new tumor. So, it is very, very dangerous. Third, this benign tumor, because of the slow growth and highly localized, it is less damaging. It shows slow growth and does not spread. That is why it is less damaging, whereas malignant tumors are very damaging. They grow very fast, they invade the neighboring cells and damage the neighboring cells. Also, they starve the normal cells by competing for vital nutrients. So, these cells which spread from to places, to distant places via the blood by a process called metastasis, They are very fatal in the sense of damaging because they grow very fast, very rapid growth. Next, not only that, they spread and damage the neighboring cells, normal cells, and also they starve the normal cells for vital nutrients. So that's why they are very damaging. So once again, the tumors formed as a result of cancer. I told you cancer is uncontrolled growth and division of the cells, and they form a lump or number of cells. This tumors are classified into benign tumors and malignant tumors. Benign tumors are confined to the original location. Second, they do not spread. And third, they are less damaging. They are less damaging because they are very slow. They grow slowly and they do not spread. Whereas malignant tumor on the other side, they are not confined or they are not localized. The cells that comes out from this tumor are carried by the blood and gets lodged to another place, distant site, and wherever they get deposited, they form a new tumor. So that process is called metastasis, by which the malignant tumor spreads via the blood and forms new tumors are called metastasis. Not only that they are spreading, they are very damaging because they grow very rapidly. They invade and damage the surrounding normal cells and also compete with the normal cells for vital nutrients. Next, causes of cancer. We can broadly classify the causes into physical cause, chemical cause and biological cause. But before that, the cancer inducing agents, what are they called? Carcinogens. So the agents that cause cancer are called carcinogens. So they can be classified into physical carcinogens, chemical carcinogens and biological carcinogens. The physical carcinogens includes the ionizing rays like X-rays and gamma rays, and the non-ionizing rays like the UV rays, which can damage the DNA and cause mutation and cancer. So physical carcinogens includes different rays, X-rays, gamma rays and UV rays. Remember, these two are ionizing rays and this is non-ionizing rays. Next, chemical carcinogens. There are a number of chemical carcinogens. One that is mentioned in your text is the one which is present in tobacco, nicotine. It's a carcinogen present in tobacco. Then you know a number of carcinogens as present in the pesticides, endosulfan present in the pesticides, etc. Then biological carcinogens, we have two important ones, viral oncogenes and cellular oncogenes. Viral oncogenes are present in oncogenic viruses. So certain viruses are oncogenic. Once they invade our body, they induce or they introduce this viral oncogenes, which brings about oncogenic transformation of the cells. Our normal cells shows uncontrolled growth and division and gets differentiated into masses of cells which are malignant. So once again, viral oncogenes which are present in oncogenic viruses. If we get infected by these oncogenic viruses, they introduce these viral oncogenes which brings about the neoplastic or malignancy, oncogenic transformation. Next, we have cellular oncogenes. Cellular oncogenes are present in the normal cells. Our normal cells have this particular oncogene, cellular oncogenes. Under certain conditions, unfortunately, they are triggered to become oncogenic. So this is evitable to anybody. Cancer. It need not be lifestyle. It need not be exposed to certain substance. Sometimes, unfortunately, our normal cells gets triggered, and this cellular oncogenes brings about, which are present in our normal cells, which under certain circumstances gets activated, can bring about oncogenic transformation. These cellular oncogenes are also known as proto-oncogenes. So once again, biological carcinogens include viral oncogenes and cellular oncogenes. Viral oncogenes are present in oncogenic viruses. We know different types of virus. So the category of virus which has this oncogenes are called oncogenic virus. If we are invaded by these oncogenic viruses, they introduce what into our body? Viral oncogenes, which brings about oncogenic transformation or which brings about malignancy. Then there is another thing, in our normal cells there is oncogenic virus, there is oncogenic gene, there is cellular oncogenes in the normal cells. Under certain circumstances, they get activated and bring about oncogenic transformations. Clear? So, once again, causes of cancer are broadly classified into three: physical, chemical and biological carcinogens. Physical carcinogens include X-rays, gamma rays and UV rays. Chemical carcinogens mentioned in the text is nicotine and tobacco, and biological carcinogens include viral oncogenes and cellular oncogenes. Viral oncogenes are present in oncogenic virus and cellular oncogenes are present in normal cells. Now, how is this viral oncogenes reaching us? When we get attacked by an oncogenic virus, then how is cellular oncogenes bring about oncogenic transformation? That is, unfortunately, due to certain circumstances, the body gets triggered or these body cells gets activated to bring about uncontrolled growth and uncontrolled cell division and cell differentiation to form these tumors. I hope carcinogens are clear. And the last template, no, second last, the detection of cancer. There are different ways of detecting cancer. Biopsy is a very common method where a slice of the susceptible tissue, the tissue which we are suspecting, is taken. A very thin section is examined under the microscope. It is first stained and then examined by the pathologist under the microscope to study the histopathology. So, that is biopsy. So, once again, biopsy, what is done in biopsy? A very thin section of the suspected tissue is taken out, stained and observed under the microscope by a pathologist to study its histopathology. Next, blood test and bone test is usually done to test the number of blood cell count, especially WBC count in the case of leukemia. Then radiography like X-rays, CT scan, MRI scan and PET scans are done to find out cancer of internal organs or the visceral organs. And we have the modern technology, molecular diagnosis called PCR. It is the latest technology which you get the result very fast, and we are going to learn this in biotechnology. It is the polymerase chain reaction where the DNA is identified and the mutation is determined and the cancer is detected. We will be learning in biotechnology. It is the latest technique which is better than the conventional technique as well as the radiography. The results are quick, and also not only the results are quick, we can identify the cancer at a very early stage. Okay, that is the advantage of molecular diagnosis, and the one is PCR, which we are hearing commonly at present for identifying COVID. So, once again, detection of cancer, biopsy, a small tissue is taken or thin suspected tissue is taken, stained, examined under the microscope by a histopathologist to find the histopathology. Then blood and bone marrow test is used to identify the number of cell counts, especially in the case of leukemia. X-ray, CT scan, MRI scan, PET scans are different radiography to find out the cancer of internal organs. And last, we have the modern method, molecular diagnosis called PCR, where the cancer can be detected at a very early stage and the result is obtained very quickly. It's faster. Now, moving on from detection to the last topic, treatment. There are mainly three treatments: surgery, radiation therapy and chemotherapy. Surgery, the particular organ that has malignancy is removed surgically so that the spread is prevented. Radiation therapy, where the particular part that is cancerous is exposed to rays, radiation, and it is lethally irradiated, taking care that the normal cells are not affected. And chemotherapy, drugs are introduced which kills the cancerous cells. But there is a drawback of chemotherapy, the side effects. What are the side effects? Hair loss and anemia. So, once again, the treatment involves surgery, where the cancerous part is surgically removed. Radiation therapy, where that cancerous region is exposed to radiation so that the cancerous cells are irradiated lethally, taking care that the cells in the vicinity, the normal cells, are not harmed. It's very careful procedure. In chemotherapy, drugs are introduced which kills the cancerous cells, but the side effects are hair loss and anemia. This is usually done in combination. So, treatment is always in combination: surgery, chemotherapy followed by surgery, and then by radiation therapy. Whatever, it is done in a combination. Usually cancer treatment involves a combination of these three: surgery, radiation therapy and chemotherapy. Now, there is another therapy called immunotherapy. Now, I brought this cancer under the immune system, not that the immune system has the ability to detect and destroy tumor cells. No, the immune system does not have the ability to detect and destroy the tumor cells. No, so it is not at all under the control of the immune system, but we can administer, the doctors administer alpha interferons. These are biological response modifiers which enables our immune system. It is given from outside. Alpha interferons are given to these patients and it enables the immune system to recognize the tumor cells and destroy them to some extent. So, it is not naturally done by the immune system, it is induced by the immune system by administering alpha interferons, and that is coming under immunotherapy. So, what is immunotherapy? The patient is administered with alpha interferons, and this alpha interferons, which serves as a biological response modifier, triggers our immune system. Our immune system is activated to respond by producing antibodies to destroy the tumors to some extent. It is not that with this the tumors are completely destroyed. This is an additional approach to bring to control the spread of tumors, where alpha interferons are administered and the immune system is triggered to respond to produce antibodies and destroy the tumor cells. This is the latest approach. So, once again, treatment involves surgery where the cancerous part is removed, radiation where the cancerous part is subjected or exposed to radiation and the cells are destroyed, taking care of the cells in the vicinity. Chemotherapy, again drugs to kill the cancerous cells. This immunotherapy where the patient is administered with alpha interferons which enables the immune system to respond by producing antibodies to destroy the tumor cells. I hope all the topics coming under this video is clear to you all. Thank you.